everyone, this is Kruti Patel from LJ Institute of Pharmacy, Department of Pharmaceutical Technology. Welcome to Pharma Ignite, the official channel of LJ Institute of Pharmacy. Today we are going to learn about different evaluations of disinfectant, or we can say evaluation methods for the chemical agents. So, evaluation of disinfectants can be mainly done by two methods. First is suspension test and second is couplet method or disk diffusion method. Now, this couplet method or disk diffusion method you are going to study, or you might have already studied in microbial assay. Okay, so couplet method is the official method of microbial assay, but that can be used for the determination of potency or evaluation of disinfectant. Okay, so in this session we are mainly going to focus on suspension test. Okay, so the suspension test is also known as or can be performed by phenol coefficient method. Now, if we look at the phenol coefficient method, then phenol coefficient can be performed by three different methods. First is RWC, that is Riddle-Walker coefficient test. Second is CMC, that is Chick-Martin coefficient test, and the third is test performed by AOSC, that is American Association of Analytical Chemists. Okay, so these three are the main methods by which we can perform phenol coefficient. Okay. So, let us start with the Riddle-Walker coefficient test. Now, if we look at the equation of Riddle-Walker coefficient test, then RWC is equals to lowest concentration of disinfectant showing growth after 5 minutes, but not after 7.5 minutes. Okay, you have to remember this equation. Divide by lowest concentration of phenol showing growth after 5 minutes, but not after 7.5 minutes. Okay, so what is the basic thing in RWC, or if we talk about phenol coefficient test, then you have to choose two substances. First will be your phenol and second will be your disinfectant. So, in this phenol coefficient test, we are going to consider phenol as most efficient, okay, most efficient disinfectant, and we are going to compare each and every disinfectant with this phenol, and we will be checking whether they are effective or not. Okay, we have certain criteria by which we can decide whether phenol is more efficient or disinfectant is more efficient. Okay, so yes, this is the formula which you need to remember. Now, microorganism used in this method is Salmonella typhi. Now, by equation you might think this is so complicated, right? But trust me, it is not so complicated. Let's start with the process. So, what you have to do, first of all, you have to prepare nutrient broth. Okay, nutrient broth you have to prepare. Then the second thing which you need to prepare is suspension or culture of microorganism. Which microorganism? Salmonella typhi. Okay, and that has to be incubated 24 hours. So, you have to take a nutrient broth, in that you have to add microorganisms and you have to incubate for 24 hours. Okay, so after 24 hours, this is the culture which you have to use. Okay. Let us look at the method first. See, in that corner there is a tube known as dilution culture tube. Okay. Then, okay, before that let me clear, by this method I will be explaining only for the disinfectant. The same part has to be repeated for phenol. Okay. So, here I have taken 4 concentration of disinfectant. Same way you have to choose 4 concentration of phenol and you have to similarly perform the test. Okay. So, yes, first of all, what I have done, I have prepared 4 dilutions of disinfectant, right? Say for example, 1 is to 100, 1 is to 200, 1 is to 300 and 1 is to 400. Okay, this is the dilution. Okay. Now, what you have to do, yes, this dilutions has to be prepared in water. Okay. So, 1 is to 100, that means 1 part disinfectant and 100 part is your water. 1 is to 200, that means 1 part is disinfectant and 200 part is water. So, as you move this way, it is diluting. Okay. So, this is how you have to prepare dilutions. Then see, in the next set, okay, I have taken test tubes, in that I have filled 5 ml nutrient broth. Okay. So, you have to fill 5 ml nutrient broth and you have to prepare 4 such sets. Okay. So, total 6 sets are there. First is for the dilutions, and rest 4 sets will be containing nutrient broth, 5 ml nutrient broth. Okay, so just learn labeling first, so it will be easier to understand, right? So first one we will be terming it as 2.5 minutes, then 5 minutes, then 7.5 minutes, and last will be 10 minutes. Okay, this is how we will be labeling our test tubes or our sets. Okay, so first is dilution set, second is you can say 2.5 minute set, then 5 minute set, 7.5 minute set, and 10 minute set. Okay, now this time set, okay, all timing sets will be containing what? 5 ml nutrient broth. Okay, chalo. Now let's start with the process. So first of all, what you will be doing, you will be transferring 0.2 ml culture. How much? 0.2 ml culture from your culture tube to all these different dilutions. You have to add it into 4 different test tubes, okay, and then you have to start counting time. Okay, so that is 0 minute, you are transferring at 0 minute. Now then you have to start counting time. After 2.5 minutes, what you will be doing, you will be transferring 0.2 ml of sample from the first set to the next one. Okay, so you have to take out 0.2 ml sample from 1:100 test tube, okay, dilution test tube, and you have to transfer it into 2.5 minute A, that is 1:100 test tube. Okay, similarly, so from 1:100 to below test tube, then 1:200 to below test tube, likewise, see I have drawn arrows. Okay, so likewise you have to transfer 0.2 ml culture. Okay, then you have to wait. Then again after 2.5 minutes, so that is total 5 minutes. Okay, after 5 minutes again you will be pipetting out sample from which test tube? From the upper test tube. Okay, from the dilution test tube, and you will be transferring 0.2 ml of your cultures or 0.2 ml of your sample again into which test tube? 5 minute test tube. Then again you have to wait for 2.5 minutes. Okay, so total after 7.5 minutes, again from the top to the third. Okay, so from the top first, top second, top third, top fourth. This is how you have to transfer. Please make sure, don't make mistake that top to first, then first to second, then second to third, then third to fourth. No, this is wrong. Don't do that. Okay, always remember why we are doing that, because we are adding microorganism into dilution of disinfectant. We are allowing them to react. Okay, so now we will be checking after what time microorganisms are getting killed. So this nutrient broth test tube, they are the media which will be growing microorganism. Okay, so by the growth of microorganism, we will be checking whether they are killed or not. Okay, so this is the reverse psychology. This is the reverse calculation. We will be checking growing microorganism, okay, and we will be checking whether our disinfectant is efficient or not. Okay, so that's why this nutrient broth, they are the media providing growth. That's why every time what you have to do, you have to transfer your sample from top set to the respective timing sets. Okay, I hope this is clear. The same way, the same method has to be performed for the phenol. Okay. After performing this work, you have to incubate all the test tubes except dilution. Okay, we don't require that dilution test tubes now. So this timing set, that is 2.5 minute set, 5 minute set, 7.5 minute set, and 10 minute set, this 4 sets of disinfectant plus phenol. Okay, this you have to incubate for 24 hours, and then next day you have to check it for the growth, whether you are getting growth or not. Now in this case, we are using nutrient broth, that's why the growth will be in form of turbidity. See, this is how you can observe. Okay, so what happens as concentration of disinfectant decreases? What happens? The growth of microorganism increases, right? So we will be having more turbidity in diluted disinfectants and less turbidity or no turbidity in case of concentrated disinfectant. Okay, so now once you get values, next day you have to keep it in equation and then you have to calculate. Okay, this is how phenol coefficient by Riddle-Walker test is performed. Okay, so let's go to formula. See, below I have written the conclusion. If RWC is equals to 1, that means phenol and disinfectant is equally potent, right? They have equal potency. If RWC is greater than 1, that means which agent is more effective? Disinfectant is more effective. Okay, and if RWC is less than 1, that means what is more effective? Your phenol is more effective. Okay, this is how you can carry out RWC test, phenol coefficient by RWC method, and you can decide whether your disinfectant is more efficient or not. Okay. Now, the next method is CMC, that is known as Chick-Martin coefficient test. Now, in contrast to RWC test, what we did in RWC, we took different samples, then we checked whether we are getting growth at 5 minutes or not, 7.5 minutes or not. Okay. So, there were 4 time slots. Now, in this CMC test, what you have to do, you have to take different concentrations, same, different concentration of disinfectant, different concentrations of phenol, you have to take. Okay. Then, what you have to do, you have to add, first of all, you have to add your sample in your dilutions, right, what we did in RWC. So, you will be adding your sample, that means you will be adding your microorganisms in dilutions. Okay. Then, after 30 minutes, you will be allowing them to react for 30 minutes, right. After 30 minutes, you will be transferring into nutrient broth, and then you will be incubating. Okay. So, your only one time transferring is there, okay, and time interval is 30 minutes. Okay. So, that is CMC. If we look at the equation of CMC, then CMC is equals to highest concentration of phenol showing growth and the lowest concentration preventing growth, divide by same for disinfectant. Okay. So, if your CMC value is equals to 1, that means again phenol and disinfectant both are equally potent. If CMC is greater than 1, that means which is more effective? Phenol. Then phenol is more effective, and if CMC is less than 1, that means your disinfectant is more potent. Okay. The more important thing which you need to remember in case of CMC is you have to add organic matter while you are preparing dilutions. Okay. We are preparing dilutions in distilled water, as I mentioned in RWC, right. First of all, you have to prepare water and disinfectant dilution, right. Same way you have to prepare dilutions here also, but the important thing is you have to add organic matter in those dilutions while preparing dilutions. Okay. What are the organic matters? You can either add 3% solution or you can add human fecal content. Now, why we are adding this organic matter into dilutions? Because it is believed, or it is not actually believed, it is proved that when organic matter are present, when foreign particles are present, they reduce the potency of disinfectant, they reduce the potency of any chemical disinfectant. Okay. So, to simulate the proper action, we are going to add the organic matter into dilution itself. So, we can have proper idea whether how much our disinfectant is going to act in presence of organic matter. Okay. So, as organic matter, we will be adding either 3% solution or human fecal content. Okay. So, let us move further. Let us look at the difference between CMC and RWC, which is very important. So, first is about organic matter. As I said, RWC that do not have organic matter, whereas in case of CMC we do add organic matter. Okay. Then next is solvent for the dilution. The solvent for the dilution in case of RWC is distilled water, whereas in case of CMC it is distilled water plus organic matter. Okay, that is 3% solution or human fecal content. The one part which I forgot is volume of medium. In case of RWC it is 5 ml nutrient broth, right? We had filled in different test tubes. So, that is 5 ml nutrient broth, whereas in case of CMC it is 10 ml. Okay. The next difference is sampling time. In case of RWC it is 2.5, 5, 7.5 and 10, whereas in case of CMC it is 30 minutes, right. The next is incubation temperature. In case of RWC it is 17 plus or minus 0.5 degrees Celsius, whereas in case of CMC it is 30 degrees Celsius. Okay. Then calculation part, you have formula, so you have to write down formula. The next is volume of microorganism. We are transferring every time 0.2 ml of culture or 0.2 ml of sample. Same is for the CMC. Here also we have to transfer 0.2 ml. Okay. Then last is incubation temperature, which is same, which is 37 degrees Celsius for both, and the incubation time, that is same again, 48 hours. Okay. So, this is the basic difference between RWC and CMC. The next method is test performed by AOAC, that is American Association of Analytical Chemists. Now, if you look at the formula, then phenol coefficient is equals to the highest dilution of disinfectant that kills microorganism in 10 minutes, but not in 5 minutes, divided by same for phenol. Your equation is similar to RWC, just the difference is instead of 7.5, instead of 5 and 7.5, here we have time duration 5 minutes and 10 minutes. That is the only difference. Okay. The other difference is that the microorganisms which are used in this case is Staphylococcus aureus or Pseudomonas aeruginosa. These two are the options. You can choose any of them. The other difference is media used. In this case, we are going to use recovery media. Now, what is this recovery media? Recovery media means media according to our disinfectant. Okay, sometimes what happens, our disinfectant, the action of disinfectant might reduce or increase in nutrient broth. Okay, so to have again proper action, we might differ different media. So we might choose recovery media. Okay, one such example is USP modified thioglycolate media, and second is lecithin medium, which is containing lecithin and polysorbate 80. Okay, so these are the different recovery media which you can use, and again as I said, sampling time would be only 3, that is 5 minutes, 10 minutes and 15 minutes. Okay. Now, the next is disadvantages of phenol coefficient method. There are various disadvantages of phenol coefficient method. First is only liquid disinfectants can be evaluated, you cannot evaluate solid disinfectants. Okay. Then second is comparison of dissimilar substances, that means you are comparing phenol and halogens, or you are comparing phenol and alcohol, which have entirely different mechanism for killing of microorganism. Okay. So yes, comparison of two dissimilar substances. Then the third is choice of test microorganism, that means we have limited choice of microorganisms, right. Some disinfectants might not effective or might not kill this specific microorganism. The third is absence of organic matter. Yes, we were adding organic matter in CMC, but not in the rest methods. Next disadvantage is long interval between successive samples. Yes, 2.5 minutes and 5 minutes. Or either 2.5 minutes or 5 minutes interval is actually long. Okay, duration to check whether microorganisms are killed at what time. That means if microorganisms are killed at 1 minute only, then what? We don't have any accountability for that. Okay, so yes, long interval, the absence of growth always cannot say that microorganisms are killed. That also means that you have not transferred microorganisms, right? So that is also again disadvantage. The lack of replication is also there. Okay, and the last is test is performed only for the one reaction temperature. Okay, we are not checking at different reaction temperature, as temperature also affects the killing of microorganisms, right. So these are the list of disadvantages. Now next is advantages of phenol coefficient method. It is of course inexpensive method and can be performed very quickly, right. The second advantage is reproducible results can be obtained, yes, but when you have experienced worker. Okay, and the last is useful to eliminate useless compounds. Okay, so this all are the advantages of phenol coefficient method. Now the next method which I had mentioned you, that is couplet method or dust diffusion method. As I said, you are going to study them in detail in microbial assay. So this is just an overview, this is just a diagram, this is how you can perform. Okay. Okay, so I hope how the disinfectants are evaluated is clear to you. For more such videos, please subscribe Pharma Ignite. Thank you so much.